Chapter 11, Part 1, or Part A, it deals with fundamentals of the nervous system and the nervous tissue. So we're going to break this down into 3 different parts. Why do you need to know this or worry about it? It's because understanding how neurotransmitters function at the neuromuscular junction, or how they function neuron to neuron, is going to help you understand how different drugs may affect the patient's nervous system. So the nervous system is the master controller, if you want. It's a communication system. We have 2 communication systems in the human body. The nervous system is one, the endocrine system is the other. The nervous system is going to respond with a very immediate but short-lived response. The cells are going to communicate by both electrical and chemical signals. So the response is very fast, very specific, but it doesn't usually last long. You have 3 overlapping functions of the nervous system. You have sensory input. So information is being gathered by various specific sensory receptors. They're picking up information on both internal and external environments, so temperature changes, calcium levels, blood pH, pain, light touch, deep pain, vibrations, sound, vision. It's picking up all of this sensory information. And then integration is where it's processing and interpreting that information. Sometimes it may interpret it and say, okay, we're aware of this, don't really need to do anything. Or sometimes it may be, oh, we need to do something about this right now. And the motor output is the activation. It's going to be your response. It's the activation of what we call the effector organs, that can either be muscles or glands, to generate some type of appropriate response. So this is just showing, okay, you see a glass, you're thirsty, sensory input, you see the glass, send that information to the brain where the integration occurs. Oh, there's a glass there, I'm thirsty, let me pick it up. How are you going to pick it up? Well, you have to send that response back down to skeletal muscle in your arm to contract, allowing you to grasp the glass and then tilt it towards your mouth. But not too fast so you don't spill it down the front, but get it to your mouth, then tilt. It's all going to be coordinated. So the nervous system is divided into 2 main parts. There are several subdivisions within these, but the main 2 parts are the central nervous system, which is the brain and the spinal cord. And this is where all the integration and the controls occur. So the sensory information is going to come here, and then the motor output leaves from the central nervous system. The peripheral nervous system, the PNS, this is everything outside of the brain and spinal cord. Now, the 2 parts are connected. So you have spinal nerves that are connected to the spinal cord. You have cranial nerves that are connected to the brain. Those spinal nerves and cranial nerves are classified in the peripheral. Yes, they're connected to the central nervous system because the 2 parts have to speak to each other, if you will, but they're not part of it, so they are with the peripheral. And this is just showing in this diagram, central nervous system, obviously your brain, your spinal cord that runs down through the foramen, through that opening of the vertebral column. And then, as you can see, your spinal nerves are attached to the spinal cord, and then you have cranial nerves that are connected to the brain. Now, the peripheral nervous system is where we're going to do additional breakdown. Think of like a flow chart of how we're separating it out. So you can divide the peripheral nervous system into functional divisions. You can divide it into the sensory and the motor. Sensory is also known as the afferent division. This is going to be even further subdivided into somatic sensory fibers, which these are receiving information from the skin, the skeletal muscles, and will join to the central nervous system. Excuse me. anything of the sensory or afferent division connect to the peripheral nervous system, or to the central nervous system. So somatic, think skeletal muscles, think skin. Visceral sensory fibers, these are taking impulses from your visceral organs. Remember, those are, think of your abdominal and pelvic organs, more of the internal organs, to the central nervous system. And then the motor division, or the efferent, this is going to be transmitting impulses from the central nervous system to the various effector organs, the muscles and the glands. That can be further subdivided into two divisions, the somatic nervous system and the autonomic nervous system. The somatic nervous system, this is where you've got motor nerve fibers that are connecting to your skeletal muscles. This is voluntary. This is what you've already seen having studied the muscular system. This would be like the motor neuron that's coming down at the neuromuscular junction. You have control over moving your hand. The autonomic nervous system, this consists of visceral motor nerve fibers, so it's going to be going to smooth muscle, cardiac muscle, and glands. It is involuntary, autonomic, involuntary. That has two subdivisions, the sympathetic and the parasympathetic. Those work in opposite aspects to each other. So like I say, think of a flow chart here. When we look at this, the central nervous system versus the peripheral. So you have the central nervous system, that's it. Now, the peripheral is the one that we can divide further. So the sensory or afferent versus the motor or efferent. And the arrows here are showing you the direction that things flow. So blue is sensory going towards the central nervous system, and the red is motor moving away. So that motor or efferent division can be divided into your somatic or autonomic, and then the autonomic can be further subdivided into the sympathetic versus parasympathetic. And we're going to be looking at all of these. But first, let's get some of the basic things down. When we look at the nervous tissue, there's two main cell types. You have your neurons, or your nerve cells. These are the ones that are specialized to be able to generate that impulse. The glial cells, or neuroglial cells, these are your supporting cells. They do not generate an impulse, but they are able to support the neurons. Now, we do have different names because there's different types of these glial cells, and we do distinguish whether they are found in the central nervous system versus the peripheral nervous system. The neuroglial cells of the central nervous system, there's 4 different types: astrocytes, microglial cells, epidermal cells, and the oligodendrocytes. The astrocytes are going to be the most abundant. They're very versatile, and they have a lot of branching to them. Sometimes they're described like these stars reaching out and touching everything. They are attached to the synaptic endings, they're attached to the neurons, they are attached to the capillaries. The brain and the spinal cord are very fragile tissues, very important tissues. They are regulating everything that is keeping you alive, your respiration rate, your heart rate, etc. It has all your cognitive functions. So obviously it needs to have an extra layer of protection. You want to have additional protection in terms of blood flow. Now, the blood, most people, okay, yeah, it's carrying oxygen and carbon dioxide. Yes, that's true. It's also carrying nutrients. Well, oftentimes it will contain toxins, and anything that you have taken into your body, drugs, etc., are often in the blood. You don't want that to come in direct contact with the brain and the spinal cord. And so the way things are set up, if you will, in the brain and the spinal cord is that with the capillaries, those are the smallest of the blood vessels. That's where all of the nutrient and gas exchange occurs between the blood and the tissues. So you basically have an extra layer, and that's going to be the astrocytes that are part of this, what we call the blood-brain barrier. Anything to come out of the blood is going to have to pass through the astrocytes before it can reach the brain. It acts like a filter, if you will. So it's helping to support the neurons in place of the spinal cord. It does play a role in this exchange between the capillaries and the neurons. It helps to control that chemical environment. It can respond to various nerve impulses and the neurotransmitters. And it just assists those neurons with keeping everyone kind of in place, if you will, and then helping with that blood-brain barrier. And this is what it looks like, kind of the star-shaped. And you can see how it's attached. It's wrapped around. This red right here is the capillary. The yellow back here is the neuron. And so you can see how it is attached to both. So it's kind of like scaffolding, but then also, like I said, anything passing out is going to have to, from the blood, will have to pass through the astrocyte first. The microglial cells, the second of the supporting cells of the central nervous system, they're very small cells. They look real thorny, like they will help migrate towards any injured type of neurons, and then they will carry out phagocytosis. So they will they help to remove any damaged extra debris. They also will phagocytize any microorganisms that were capable of penetrating and getting into the central nervous system. So they're for protection and also cleanup crew. And that's what these are. This is kind of like going in a real sticky briar patch or something. The epidermal cells, these can range in their shape from squamous to columnar. Some of them are ciliated. Those that are ciliated, the beating pattern that you tend to see with that helps to circulate the cerebral spinal fluid, or CSF. These cells tend to line the central cavities of the brain, the spinal cord, and they are there's certain areas where they tend to congregate in the choroid plexus, which is basically responsible for producing the cerebral spinal fluid as well. So here you can see these cells with the cilia, and you can see as the cilia moves, it's going to be moving this cerebral spinal fluid. It's like, keep on moving. The oligodendrocytes, these are cells that are branched. They will be wrapping around the axons of the neurons, basically helping to provide insulation. They form this myelin sheath, and it provides that insulation, which allows the electrical impulse or that action potential to move much faster. It's almost, think of if any of you are familiar with electricity, which I'm sure many of you are more familiar with that than I am, but it's kind of like having your wire and then having the insulation wrapped around that copper wiring on there. Now, with the peripheral nervous system, there's only two kinds of the neuroglial cells, or supporting cells: satellite cells and Schwann cells. The satellite cells are very similar to the astrocytes that you saw in the central nervous system. And then the Schwann cells in some ways are similar to the oligodendrocytes in that they will also form this myelin sheath, giving that insulation. And the Schwann cells are going to be very important with the peripheral nerve fibers when they are damaged. The Schwann cells are going to help direct them during the regeneration process. So they're very important for that. The nerves in the central nervous system do not regenerate. So the Schwann cells, as you can see here, along the axon, and the satellite cells up here. So the neuron is the functional unit because it's producing that impulse, that electrical charge. It receives a stimulus and it will travel down the length of the neuron and then be able to stimulate or activate or innervate another neuron or a muscle or a gland, whatever its connection is. Certain characteristics of neurons is that usually they will last a person's lifetime. They usually do not carry out mitosis. They have a very high metabolic rate, so they need a very good, rich supply of oxygen and glucose. They do have what's known as a cell body. That's where the main portion of the cell is. That's where the nucleus is going to be and most of the organelles. And then you're going to have extensions or processes off from that. The cell body is also called the soma. Some books you may see it called the pericaryon. You do have, like I said, a lot of your organelles, such as your Rabies there, a lot of your ribosomes for making proteins. You have your nucleus there. There may be some pigmentation there. And then a couple of terminologies that we use. When you have a cluster of these cell bodies, we tend to give them different names, whether they're in the central nervous system or whether they're in the peripheral nervous system. Most of the neuron cell bodies actually will be located in the central nervous system, and they're going to be called nuclei. Now, this is not the same thing as the nucleus of a cell, or the same thing as when you're talking about an atom and the nucleus of the atom. Unfortunately, it's a similar name, but it has different meaning. So it's just a cluster of cell bodies in the central nervous system. Now, if that cluster of the cell bodies is in the peripheral nervous system, then we call it the ganglion. So if any of you have heard of a ganglion cyst, that tends to be where you have a cyst at a cluster of these cell bodies in the peripheral nervous system. So you have these different processes extending from the body. Now, some of them are very short, known as the dendrites, and then the very long extensions are the axons. And the axon is carrying the electrical impulse away from the cell body. In the central nervous system, once again, you have a lot of the cell bodies there and the processes. Peripheral nervous system is mostly these processes. When you have a bundle of axons in the central nervous system, we call it a tract. In the peripheral nervous system, when you have a bundle of the processes, we call it nerves, which is kind of a confusing term, but like when we talk about cranial nerves, spinal nerves, it's a whole bundle of these neuron processes, usually the axons. The dendrites, as I mentioned just a bit ago, are usually much smaller, and they are sending impulses towards the cell body. So in this picture, you can see in this large yellow area here, this is the soma or the cell body. You have a nucleus in here, you've got your mitochondria, you've got your Golgi apparatus, your Rabies, all those other organelles are in here. These smaller processes or extensions, those are the dendrites. They will be picking up a stimulus and sending that impulse towards the soma. So any impulse picked up is going to be coming towards the cell body from here. Now, notice this process is much longer than the dendrites. This is the axon. So on the axon, the impulse, that electrical charge or impulse or action potential, we're all talking about the same thing here. It is going to be moving away from the soma towards the axon terminus. And this is what is then going to synapse or connect to either another neuron, a gland, or a muscle. Where the axon leaves the cell body, notice it kind of looks like a funnel shape right here. This is known as the axon helix area. And then this is with a peripheral nerve because you've got the Schwann cells here that are providing that insulation. The space between them that is not got insulation is known as the node of Ranvier. So those dendrites, they're very short. The numbers of those are going to vary. They're sensory in the standpoint they're receiving the input. They're going to send that message towards the cell body. Sometimes these may have spiky ends on them. Some of them may have little swellings. It just depends on which one you're talking about. So in this particular picture right here, here's the neuron, and you've got the dendrites extending out. Okay. With the axon, as I said, where it leaves, it's kind of that funnel shape. That is the axon helix. How long is an axon going to be? It varies. Some are short, some are very long. The longest ones are about a meter. For those of you not familiar with the metric system, a meter is a little bit over 3 feet. So you have some that go from your spinal cord all the way down to your toes. Long axons, the bundles of them are known as nerve fibers. And at the end, we call it the terminus, and that is what is going to connect to, like I said, another nerve, be involved with the neuromuscular junction or with the gland. So the action potential is moving down the axon, away from the soma. The neuron cell membranes referred to as the axolemma. At the terminus, you do have those neurotransmitters in the vesicles. The axons are going to rely on various proteins and membranes. If it is cut, it is going to very quickly decay, and depending on the location, it depends on the location, and you have to look at the individual circumstances as to whether or not it can be regenerated. Now, in terms of some of the clinical applications, when you're talking about the individual neurons, disorders, and diseases, different viruses and sometimes toxins from bacteria and fungi can damage the neural tissue. It'll depend on the specific organism as to what type of toxin it's producing, as to what type of damage will be done, or the specific, say, virus and what type of damage you see. Most of the time, things are moving from the soma down the axon, so it's moving away from the soma. Retrograde flow means it's flowing the opposite way. It's flowing from the axon back towards the soma. And some toxins take advantage of this, some viruses take advantage of that. Rabies. Rabies is caused by a virus. It is able to use the retrograde flow in order to cause the damage. What it will do in the infection, what happens is a person is bit by an animal that has rabies, and what will happen at that point is you're bit. The virus enters because of the broken skin on the peripheral nerves. The virus is able to move up the neuron by that retrograde flow, up to the soma, up those dendrites. So it's able to move essentially from the peripheral nervous system to the central nervous system. And eventually it moves all the way up to the brain. Just rabies, just for your own information, is kind of historically an interesting virus to study. Just so you know, by the time an individual shows symptoms of rabies, it is too late to treat. The classic symptoms are you tend to have a lot of saliva production, and it becomes very painful actually to drink. So you get very thirsty. When you're thirsty, you produce more saliva. So that's why you have that excess saliva production. And like I say, you're very thirsty, but it physically hurts to drink. So you don't drink, so you produce more saliva, which causes more pain, and um, it breaks down the brain tissue. It destroys the brain. Um, but once you show those symptoms, like I said, it's too late. There's, in all of recorded history, I can remember taking a class and studying about rabies, and it was stated that in all of recorded history, there have been only 5 confirmed cases of where someone was diagnosed with rabies, where they showed the symptoms and they survived. Those odds are not very good. Now, obviously, there are a lot of people who you could tell stories about being bitten by an animal contracting rabies. Well, we have shots now that you can administer to the person that prevents them from getting the disease. So there's a difference. In microbiology, we often talk, there's a difference between being infected and having a disease. The problem with the rabies, if you are bit by an animal, you need to start the shots immediately. If you do not have the animal, you need to start the shots. If you do have the animal that bit you, they can test the animal to see if it does have rabies. Now, you can either observe it, but that's kind of taking a chance, because if you wait for that animal to show symptoms or not, you're kind of playing Russian roulette here in that, well, if you have rabies, you might show symptoms before the animal does that bit you. The only way to conclusively diagnose it is they have to look at the brain tissue, which means they have to sacrifice the animal. But that's kind of the classic example of a virus that will travel by retrograde movement. And how did you get it? Because of the bite, and the virus was injected at the bite site. There is some research wondering if this is a way that we can use to potentially treat some genetic diseases. So it's being looked at. The myelin sheath, that covering around the axon, is composed of a substance that has both protein and lipids in it, and it does look white. Like I said, it's going to insulate the axon. By insulating it, that's going to increase the speed that impulse can travel down that myelin. When you look at fibers, those axons that are myelinated, you will see that if they're myelinated, they have a whitish color to them. The non-myelinated ones do not have that sheath. They do not look white, and the impulse is going to travel much slower. So that myelination, as I mentioned earlier, is made or formed by the Schwann cells. They're going to wrap around that axon. Now, one cell forms a little segment. It's not going to cover the whole thing. So the entire length of the axon is going to have multiple Schwann cells wrapped around it. You don't have to worry about the exact process, how it forms, but to wrap around. See, it's kind of like, it's described like a jelly roll. It's like you start here and you just keep twisting around. The layers get thinner and thinner, but you're covering it until it is wrapped around. And this is a cross section, so here's your axon. So you can kind of make out that there's different layers in there of the myelin sheath. But that's all one cell, but it's been tightly wrapped around. There are gaps between the cells. They are known as the nodes of Ranvier. I pointed them out in a previous picture. You're going to see how that's going to play a role in the way that the electrical charge will travel down the axon. So here, once again, is the node. So this is a cell, this is a cell, this is a cell. And between each one, you've got those nodes. In the central nervous system, remember, it's the oligodendrocytes that form this myelin sheath. One cell, they're much larger, one cell can wrap up to 60 axons at once. You do have these gaps present. In the central nervous system, when we move on and study the brain and the spinal cord, you'll notice that we'll talk about white matter versus gray matter. Well, the white matter, those are areas where you have a lot of myelinated fibers because that lipid gives it a whitish color. So you know the fibers are myelinated. The gray matter is going to be mostly cell bodies as well as non-myelinated fibers. So it doesn't have those lipids, so it doesn't look white. And it literally is a color difference between them. This is showing the oligodendrocytes forming the myelin sheath in the CNS. You can classify neurons by structure and by function. Structural classification, there's 3 types: multipolar, bipolar, and unipolar. Multipolar, as the name implies, you have 3 or more processes. Usually you have a lot of dendrites and 1 axon. This is the most common type that you will see of a neuron in the central nervous system. Bipolar, and this is where you have 2 processes. You have 1 axon and you have 1 dendrite. This, probably the most common place where you'll find a bipolar neuron is going to be in the retina, which is in the eye. And then unipolar, you have basically 2 axons is what it looks like. So here we have the cell body in the multipolar with multiple dendrites and 1 long axon. So you have these multiple processes here. In the bipolar, you have a cell body, and on one end you have the dendrite, the other end you have the axon. So you just have the 2 processes off of the cell body. On a unipolar, it's like you have just 1 long process and the cell body is off to the side. See how the bipolar, the cell body is in between? But on the unipolar, it's off on the side. And this just gives some examples of additional structures, variations of what they may look like. In terms of functional classification, they're either sensory or they're motor. There's also an interneuron that you will see. First, starting, you have sensory. That's going to be transmitting the impulses from receptors towards your central nervous system. Most of these are going to be unipolar ones. The motor, it's going the opposite way. It's sending impulses to the effector, so it's going from or away from the central nervous system. And then the third type are your interneurons. Some books will call these your association neurons. They're going to be the intermediate. They're in between the sensory. So the sensory neuron is going to stimulate the interneuron or association neuron, which is then going to stimulate the motor neuron. Most of these are going to be found within the central nervous system. So the peripheral nervous system picks up the sensory, sends it to the interneuron in the central nervous system, which then interprets all the information, sends the impulse down the motor neuron of the peripheral nervous system. And this is showing additionally the comparisons, once again, between some of these. So you can classify both by structure or by function. So this portion of Chapter 11 is just looking at some of the histology and setting the groundwork, where then we're going to go into, in the next part, go into more detail with the nervous system.